Hello, College Biology. This is Shipping and Receiving, Chapter 2, Exocytosis. And so if we dissect this word, exocytosis, we know that it is a process. We know that it has to do with cells, and exo means outside. So this is a process of getting things outside the cell. And this story starts with our production story. Basically, we've got our chromosomes and our nucleus that get copied into mRNA, and that mRNA is taken out of a nuclear pore, and then eventually gets to a ribosome. So in this model, the ribosomes are the little purple dots, and they are connected to the endoplasmic reticulum in this case. That's not always the case, but since we're about shipping and receiving, we're going to involve the endoplasmic reticulum. If the mRNA gets to a ribosome that's just floating around in the cell and it's not in the cytoplasm and it's not attached to the endoplasmic reticulum, the protein will get made and then it will float around in the cytoplasm until it encounters something of significance. In this case, when the mRNA is accepted by a ribosome that is on the endoplasmic reticulum, it immediately gets, that protein product immediately gets processed by the endoplasmic reticulum. So what does that mean? What happens? Well, this area is putting finishing touches and edits on protein that are being made before packaging them into transport vesicles. A transport vesicle, we've talked about vesicles before, specifically denotes that this vesicle is has the ability to transfer things within the cell, but it cannot fuse with the cell membrane and it cannot leave the cell. So the endoplasmic reticulum can't prepare things to exit the cell. That is outside of its scope, outside of its ability. Now the smooth ER, well, if there are no ribosomes there, then there's no protein products that are going to get made. So the smooth ER really doesn't deal much with proteins. That part is used to make phospholipids that will be used in other parts of the cell. So we have lots of membranes in the cell. We've got a cell membrane, we've got membranes that are part of the Golgi body, we have membranes that are part of the ER, we have membranes that are part of the nucleus, we have membranes that surround other organelles. So we need phospholipids, and they got to be made someplace. So they're often made in the smooth ER. So we've got protein products made in the rough ER and packaged. We've got lipids made in the smooth ER and packaged, and either way they are packaged in transport vesicles. Now a transport vesicle can travel to the Golgi body, where the Golgi body can modify that vesicle. And one of the things that it can do is it can create a secretory vesicle, because it's going to be secreted from the cell. These vesicles have been modified so that they can fuse with the cell membrane and release their contents to the outside of the cell. So the Golgi body creates the secretory vesicles for external purposes. Now, the Golgi body also makes lysosomes. If the protein in the transport vesicle is a set of digestive enzymes, then the Golgi body will kind of put some finishing touches on that vesicle and turn it into a lysosome so that it can digest things in the cell. So that's sort of the story. Those are the things involved. We can take a look at this. You've seen something similar to this before. Exocytosis is very similar to endocytosis. It's just the reverse process. You have your vesicle. It fuses. It splits open and fuses with the cell membrane, and then it releases its interior products to the exterior of the cell. That is our shipping story. I hope this helps. Struggle well.